A case study on prostate cancer by Scott Brennan. Cancer is a disorder of altered cell growth and differentiation. Neoplasms tend to increase in size and often continue to grow after initial stimulus is gone. Unlike normal tissue growth, neoplasm growth is unstructured and out of control. There are two types of cancer. Benign tumors remain localized to their site of origin and lack the ability to metastasize, infiltrate, or invade other sites. Malignant tumors invade and spread to other parts of the body. They have a greater potential to grow rapidly and widely. In order to understand cancer, knowledge of the cell cycle is essential. The cell cycle is an orderly sequence of events which occurs during cell proliferation and differentiation. During the cell cycle, genetic information is duplicated and distributed between two identical daughter cells. There are four distinct phases of the cell cycle. Within the cell cycle, there are a number of sequences in place to ensure that all phases of the cell cycle have taken place. Normal healthy cells go through this cell cycle in a normal behaving manner known as mitosis. Cells receive signals when to grow and are usually located in a generalized area. Any cells which don't follow the sequence of the cell cycle are destroyed by a process known as apoptosis. However, with continued replication and proliferation, a cell may develop the ability to avoid anti-growth signals and then further down the track avoid apoptosis. In turn, they will then continue to grow at rapid and uncontrollable rates, and this is when cancer develops. This is most likely why most cancers are developed later in life, as the cell cycle has gone through a number of times and these cells have had greater chances to develop multiple mutations. The prostate contains many small glands. In prostate cancer, the cells that are produced by these glands mutate into cancerous cells. It is therefore that prostate cancer is considered to be a glandular cancer. The prostate glands require male hormones known as androgens to work correctly. These androgens include testosterone, dihydroepiandrosterone, and dihydrotestosterone. The region of the prostate gland where the endocarcinoma is most common is the peripheral zone. Initially, small clumps of cancer cells remain confined to the otherwise normal prostate glands. However, over time, these cancer cells may begin to multiply and spread to the surrounding tissues of the prostate, also known as a stroma. Here, a tumor will be formed. Eventually, this tumor may grow large enough to invade nearby organs such as the seminal vesicles, the rectum, or the tumor cells may develop the ability to travel into the bloodstream or lymphatic system. Prostate cancer is considered to be a malignant tumor because it is a mass of cells that can invade other parts of the body. The invasion of other organs is called metastasis. Prostate cancer most commonly metastasizes to the bone, lymph nodes, and may invade the rectum and bladder after local progression. The route of metastasis to the bone is thought to be venous. 85% of men who die from prostate cancer have bone metastasis present. There are high amounts of blood flow through the bone marrow, which gives malignant tumors ample access to the bone. Prostate cells express adhesion molecules with affinity for the bone. It is therefore believed that once the tumor has settled and attached, it is able to produce its own growth through local growth factors. Furthermore, as prostate cancers secrete endothelium-1, they promote their own growth through the stimulation of osteoblasts. The causes of prostate cancer is quite complex and still poorly understood. However, it is believed to stem from some type of chronic inflammation, trauma, genetics, or sex hormone influence. It has been well documented that chronic inflammation leads to a number of human cancers, including prostate cancer. Chronic inflammation seems to affect the pathogenesis of cancer by causing damage to the DNA in cells, triggering the proliferation of these damaged cells, and creating an ideal environment which enhances cell replication. Both normal healthy prostate cells and malignant cancerous cells rely on androgen stimulation for growth and proliferation. While testosterone is not considered to be the cause of prostate cancer, it is believed to play an important role in the growth of these tumors. The balance between androgen and estrogen levels is essential for maintaining homeostasis and good prostate health. Research has suggested that if these two hormones become unbalanced, then the risk for malignant and benign diseases increases. In a healthy prostate, the aromatase enzyme is contained within the stroma. However, in a malignant prostate, the enzyme is active within the epithelial. This has been linked to the development of prostate cancer. Interestingly, epidemiological studies have found little evidence to support the association between sex hormones and prostate cancer. There are 4 stages used by health medical professionals to determine the level of prostate cancer. The stage of prostate cancer depends on several factors, including whether the prostate has spread to nearby lymph nodes or other parts of the body, whether the tumour has invaded nearby organs, the Gleason score, as well as the PSA level. In stage 1, the tumour remains contained inside the prostate. It may also be too small to be detected by a digital rectal exam. In stage 2, the tumour is slightly more advanced than stage 1, however it still remains contained within the prostate and has not spread. In stage 3, the tumour has now spread beyond the prostate. It may have possibly invaded nearby small vesicles. Cancerous cells, however, have still not spread into the lymph nodes. In stage 4, the tumour may have now spread to other nearby organs, including the bladder, rectum, or other nearby structures. It may now have also spread to the lymph nodes, bones, and other parts of the body. The prostate-specific antigen, which will be referred to as PSA, is a glycoprotein enzyme which is secreted by the epithelial cells of the prostate gland. A PSA test quantifies the level of PSA in the blood and has been established as a marker specific for prostate epithelium. A positive PSA test only indicates the possible presence of prostate cancer, as elevated PSA levels can also be the rise of other prostate conditions such as benign prostatic hyperplasia and prostatitis. There is little evidence to suggest that either of these conditions will lead to prostate cancer. There are several limitations to the PSA test. A PSA test may give either false positive or false negative results. Most men who have elevated PSA levels turn out to not have prostate cancer. Only about 25% of men who have a prostate biopsy following high PSA levels turn out to have prostate cancer. One must be careful when using the PSA test to diagnose prostate cancer, particularly in the early stages of the disease. As research has shown, people with early stages of prostate cancer, PSA levels may actually be below 20 nanograms per litre. A biopsy is therefore recommended to determine the presence of cancerous cells. During a biopsy procedure, several samples of prostate tissue are taken. The biopsy sample is analysed by urologists who will determine if there is any cancerous cells present. There is no defined normal level of PSA. However, anything less than 4 nanograms per milliliter is determined to be safe. PSA levels may be important in determining the stage of the disease as well as helping manage it. A correlation has been identified between the levels of PSA and the stage of disease, as shown in the figure above. The initial baseline testing of PSA can be used to determine changes following treatment. PSA levels should return to zero following a treatment. Increases in PSA levels may indicate the reoccurrence of disease. So what does this mean for David? David's initial serum PSA level of 32 nanograms per litre places him into a higher risk category for having prostate cancer. A biopsy is therefore needed to be confirmed whether David actually has prostate cancer or not. If prostate cancer is present, it may be only localized. Radiation therapy is used to destroy and kill cancer cells. In prostate cancer, a greater number of prostate cells are produced. As these cells produce prostate-specific antigens, this is why we see a higher amount of these levels. Following radiation therapy, the prostate cells become destroyed, and therefore we see a decline in the prostate-specific antigens as a result of the destruction and death of these cells. As shown in the figure above, prostate-specific antigen level drops significantly following radiation therapy. 3 months post-radiation therapy, the prostate-specific antigen levels dropped from 12.5 nanograms per milliliter to 2.6 nanograms per milliliter. Huggins and Hodges observed that the growth of prostate cancer is dependent upon the presence of androgens. Testosterone suppression and deprivation by estrogens have become the ideal treatment for prostate cancer. However, due to the significant side effects of these treatments, a new approach was needed. In 1971, Scully and colleagues discovered the luteinizing hormone-releasing hormone, which led to the development and use of the luteinizing hormone-releasing hormone agonists. Luteinized hormone-releasing hormone is secreted by the hypothalamus. It travels to the anterior pituitary gland where it binds to its receptor. This leads to the stimulation and production of the luteinizing hormones and follicle-stimulating hormones. Circulating luteinizing hormones enters the systemic circulation where it interacts with the receptors of the Leydig cells located within the testes. This interaction leads to the production of testosterone. 90% of circulating testosterone is bound to either the sex hormone binding globulin or to albumin. The other 10% is unbound and available for cellular activation. Testosterone levels are regulated by testosterone itself, where if there is too much circulating testosterone, it inhibits the release of its production by inhibiting the luteinizing hormone releasing hormone from the hypothalamus. Testosterone, which is produced by the Leydig cells, is transported to a number of organs through systemic circulation, including the prostate. Inside the prostate, testosterone is converted by an enzymatic action to dehydrotestosterone. It then binds to the androgen receptor on the cell nucleus, where it finally results in the synthesis of prostate-specific antigens. Inside the prostate cancer cells, dehydrotestosterone binds to the androgen receptor. The androgen receptor complex binds to the corresponding androgen receptors in the element of DNA. This leads to the stimulation and activation of transcript and translation of genes, and then further causes prostate cancer tumor growth. Approximately 70% of prostate cancers are testosterone dependent, whereby the survival and growth of the cancerous cells is regulated by the androgens. It is for this reason that treatment for patients with advanced stages of prostate cancer is aimed at the suppression of these androgens. During therapy with luteinizing hormone releasing agonists, there is an initial increase in the luteinizing hormones produced from the pituitary gland. Consequently, this will result in a surge and production of testosterone levels above normal values. Although this initial surge of testosterone production, continued treatment with luteinizing hormone releasing agonists leads to the desensitization and downregulation of its receptor, which subsequently leads to the inhibition of the luteinizing hormone and thereby a reduction in testosterone levels. One downside to this therapy is the possibility that cancerous cells may develop a way around being androgen dependent. It has been suggested that prostate cancer cells adapt to androgen therapy by increasing gene expression and androgen biosynthesis and the activation of other pathways which can enhance tumor growth. There are a number of musculoskeletal side effects which accompany the long-term use of the luteinizing hormone releasing hormone agonists as a therapy. These musculoskeletal side effects of hormone therapy include osteoporosis, loss of muscle mass, and loss of muscle strength. Osteoporosis is an important problem in patients who receive long-term androgen therapy. Luteinizing hormone releasing hormone agonists downregulate the anterior pituitary gland receptors and thereby decreasing serum testosterone to castration levels. Androgens play an important role in bone metabolism. Osteoblasts have androgen receptors which influence them in a number of ways, including growth and proliferation, bone matrix production, and cytokine production. The reduction of testosterone levels induced by LHRHA therapy, also referred to as hypogonadism, has shown to have a significant effect on bone mineral density. A decrease in bone mineral density is observed following this type of treatment, which is often associated and leads to a heightened risk of bone fractures. A 14% incident rate of bone fractures in men with luteinizing hormone releasing hormone agonists as treatment is shown when compared to a 1% in a control group. Furthermore, a 5 to 10% loss in bone mineral density has been observed. Another issue for patients who use hormone therapy as a treatment for prostate cancer is the progressive loss of muscle mass. As discussed previously, testosterone is converted to dehydrotestosterone, which is found in skeletal muscles. As hormone therapy leads to castration levels of testosterone, it could very well be possible that muscle loss from this type of therapy may be closely related to the significant reduction in testosterone levels. Moras and colleagues found that following short-term androgen deprivation therapy, a reduction in protein synthesis is evident. Protein synthesis is one of the predominant factors in muscle hypertrophy. Additionally, testosterone leads to a cascade of events which indirectly affects the skeletal muscle cell. Arguably, the most significant influence it has is the effect it has on the insulin growth factor and growth hormones. Both of these are anabolic hormones where insulin growth factor stimulates muscle protein synthesis and in turn, muscle hypertrophy. A study by Boxer and colleagues examined the effects of the luteinizing hormone releasing hormone agonist treatment and the effects that it had on body fat percentage and muscle loss. Their study showed a significant increase in total body fat percentage. This was attributed to a loss of skeletal muscle mass and an increase in adipose tissue following the treatment. Strength loss is also another issue for patients who use luteinizing hormone-releasing hormone agonists as a treatment for prostate cancer. This type of treatment seems to have a significant effect on the neuromuscular junction. A relationship between the dose of luteinizing hormone-releasing hormone agonist and the acetylcholine receptor has been found. Higher doses of treatment has been associated with greater desensitization of the acetylcholine receptor. Therefore, it can be assumed that with supraphysiological amounts of therapy, as seen in patients who receive this treatment, a decreased polarization across the postsynaptic clefts can be seen. This can potentially lead to decreased firing rates of action potential, which is essential for the contraction of skeletal muscles and can ultimately cause muscle weakness. Furthermore, loss of muscle strength can also be directly related to muscle atrophy, as already discussed previously.